My Verizon tower tells me it's start time, so let us begin. Welcome and good morning. Welcome to what has become an annual Harmful Algal Bloom Research Symposium. The Algal Bloom Action Team is thrilled to be hosting this event, our third, and we are glad you can join us. This year's symposium is organized into 4 sessions spanning 2 days. Today's session will focus on harmful algal bloom or HAB monitoring and forecasting in the morning, HAB detection and treatment in the afternoon. Friday's sessions will focus on ecology and human health and case studies of effective or innovative bloom management. My name is Eugene Bragg. I am Program Director for Aquatic Ecosystems Extension at The Ohio State University. I'll be your moderator for the morning session. Each session will be facilitated by a member of the Algal Bloom Action Team, a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting this annual symposium, our team is developing a website of resources that can be used freely for outreach and education around harmful algal blooms in the North Central region. I encourage each of you to visit that page. That is at northcentral.org/HABs, which will be placed in the chat, I believe, by Ann. I hope by Ann, because I'm not typing. There you can explore some of the resources we have to date, including a what you should know fact sheet, a fact sheet on prevention and treating harmful algal blooms for landowners, and a HABs frequently asked questions database. In addition to this symposium, we also host a bi-monthly webinar series where we feature the latest harmful algal bloom research year-round. On our website, you can find recordings of all our past webinars, information on upcoming webinars, as well as access information and recordings on our past HAB research symposia. Before we get started, a few housekeeping items. We'll have a 30-minute keynote presentation from Heather Raymond of my very own Ohio State University, followed by a 10-minute question and answer session, followed by 4 20-minute presentations with a 20-minute break in between the third and fourth talks. We will allow time for questions and answers after each presentation. Have your questions addressed during this time. Please post them using the Q&A function, the panel, which can be located at the bottom of your Zoom screen. If there is a question already posted to the Q&A panel and you have the same question, use the upvote function on the similar question to help ensure we get answers to the most popular questions during the panel. Typically, we have more questions than we can address, so please upvote those questions so that we address the most popular ones first. Please note that the Q&A panel function is separate from the chat function in Zoom. Again, questions for the presenter should be posted to the Q&A panel. If you are having any technical difficulties during the symposium, please let us know via Zoom's chat function. The chat function will be for those technical problems, and we will be happy to assist you as best we can. You can also use the chat function to introduce yourself or discuss the research findings with fellow water professionals at the symposium today. Following the morning session, we will break for lunch and resume at 1 p.m. for session 2. All symposium presentations are being recorded and will be posted to the Algal Bloom Action Team website at the conclusion of the event. We will also post the presentation slides to this website for your reference. Lastly, before we get started, we'd like to thank our symposium sponsors. Special thanks to our support sponsors, Utrofix and Limnotech, and for our partner sponsor, Flowcam, for making the event possible today. So let us jump right in. We are thrilled to have Heather Raymond with us today as our keynote speaker to discuss harmful algal bloom control resources and innovations. So thank you, Heather. Heather is a colleague of mine. You can read a brief bio of her on the screen as we speak, but I want to get right to the presentation. Welcome, Heather, and thank you for joining us.
Thanks, Eugene. I'll go ahead and start. It says I cannot share my screen while the participants are sharing, so let me
try now. I've just stopped the introduction presentation.
Perfect. Okay, are you seeing that correctly?
I'm seeing your view, the presenter's view.
Oh, no. When we practiced this 2 times, you were not seeing the presenter's view. Let's see how we can edit that.
Go to display settings at the top and click that down button. You should be able to switch real quick.
Display settings at the slide control, or
Yeah, at the top there. Yeah, there. Otherwise, you can stop sharing and try it again as well.
let's just do that. Better?
Still seeing your presenter's view.
Okay, so if I do which should I select? Hide video panel or?
Yeah, so at the top it says show taskbar display settings or in slideshow. If you click the down arrow for display settings, you should be able to swap the two.
Okay, is that on my PowerPoint?
Yes, move further.
Let's see. We'll try this one more time. Okay. This is what happens, man. You try 3 times and it works perfectly.
Well, the third was the charm. All points
Yeah.
beyond that are, no, all bets are off.
The third was the problem, right? Okay, let's go here. Let's do share. Let's go here. And
That looks more like I'd expect.
still looks good.
Yes.
Okay, I'll go without notes. We'll be fine. Okay, thank you again. I'm excited to be here today to talk about harmful algal bloom control resources and research and innovations. This isn't really aligned with this session's theme, but it's more kind of overarching for the whole symposium. I'll be covering control strategies, or those strategies that focus on direct treatment of cyanobacteria harmful algal blooms. I do not have time to discuss HAB prevention. I know that's been the focus of other previous webinars, but we do recognize that prevention strategies are also important. Just to know we're focusing on control today. I'd like to start by directing you to an excellent online resource developed by the Interstate Technology Regulatory Council, or ITRC. The link to all of their guidance documents, including HAB monitoring, response planning, and case studies, is included at the top. And a direct link to their guidance on harmful algal bloom management strategies is highlighted in yellow. The ITRC spent several years collaborating with experts in the field on this comprehensive guidance and associated HAB management strategy selection tool. The guidance includes an alphabetical table of management strategies with a brief description of each strategy, relative cost, applicable size and type of water body, and if the field data supports the effectiveness of the strategy. More detailed information on each of these individual strategies is available within the links that are highlighted to the left, including references and peer-reviewed articles with supporting information and some case studies. Now, if you're a lake manager and you're watching this and you've worked with a company that is guaranteeing their control strategy will solve your problem, I would recommend you look at the available data on this specific strategy here. And I'd also stress that no strategy is perfect and will be effective in every situation. Now, since I'll barely have time today to go in depth on even one new innovative strategy, I suggest that those interested in learning more about HAB control options, check out this resource. It's excellent and is a great starting point. I also want to quickly point out that the ITRC management criteria tool shown here, using this tool, you can filter the strategies that are in that list based on whether or not they're focused on management intervention, which is also another name for control, or more on the prevention. It also can be filtered on the type of water body and whether or not you're dealing with a planktonic or within the water column type of bloom, or a benthic bloom, which is on the bottom or attached to surfaces. I included this slide mainly because if you click on the selection tool link in the prior link that I provided, it defaults to the monitoring tool. So it's kind of confusing when you first go there. So to find the actual management tool, when you click on that link on the left sidebar, there's this interactive tools section, and you'll want to click on the green management criteria tool to go where you're needed. I've contacted ITRC about fixing that default link, but hopefully this will save me from some "why isn't this working" emails. Another forthcoming document is the harmful algal research and response, the CADL National Environmental Science Strategy. I helped work on this. It's currently drafted, should be available soon. The National HAB Committee solicited input from a broad group of HAB researchers and practitioners to update the last CADL strategy. This strategy includes a section on harmful algal bloom control. Freshwater and marine HAB control strategies are categorized based on available effectiveness data, and primary data gaps that need to be addressed are identified. The scientific consensus strategy is currently undergoing final edits and should be available to the public soon. So stay tuned for that. I'm sure there'll be some broad announcements, and we can contact this group when it becomes available. I would like to quickly point out that out of all of the control strategies, only 3, algicides, phosphorus binding agents, and to a more limited extent, shading compounds, were identified as well-developed strategies with demonstrated effectiveness at HAB control. Many additional strategies are identified as promising, but needed more lab and field scale data to confirm effectiveness. So we really have a long way to go. Thankfully, given the need for effective HAB control strategies, multiple federal and state agencies are committing resources to help fill those HAB control data gaps. PCM-HAB is authorized as a national competitive research program within NOAA under the 1998 Harmful Algal Bloom and Hypoxia Research and Control Act. Say that 10 times fast. It's also called HABARCA for short because of the long name. That was reauthorized in 2014 or 2004, and then again in 2014. However, NOAA's jurisdiction is marine and Great Lakes coastal waters, including Lake Erie and the other Great Lakes. But it does not extend into inland lake HAB control. However, some of the project data is transferable to inland waters and can be useful. Examples of funded projects include use of clay flocculation to control Karenia brevis marine dinoflagellate blooms, and using microsystems degrading bacteria and their enzymes for drinking water treatment. So that is one of the potentially transferable strategies. Now, NOAA recognized the need for more effective HAB control strategies, and last year they funded a new HAB technology incubator that is being run through a cooperative agreement with IMET at the University of Maryland and the Mote Marine Lab out of Florida. The incubator will provide seed funds for research on both marine and freshwater HAB control strategies. I'm personally hoping for an RFP in 2023 this year. The incubator is also tasked with developing a clearinghouse of relevant permit and environmental compliance requirements that may be useful to anyone here interested in conducting HAB control trials. As someone who's conducted control trials myself, the federal and state permitting process can be pretty burdensome. So I'm hoping that this will be useful, a useful tool for many folks involved. U.S. Army Corps of Engineers has jurisdiction over Corps-operated inland lakes, and through their Aquatic Nuisance Species Research Program and recent targeted appropriations, they've supported both internal and extramural HAB control applied research projects. Many of the innovative projects they're supported were highlighted at the last U.S. Symposium on Harmful Algae in New York, including a harmful algal bloom interception, treatment, and transformation system that is based on dissolved air flotation and biomass conversion to biofuels. This one's pretty exciting. There's a couple other companies, AEOCOM, that are looking at this kind of cradle to grave treatment approach. They also have some innovative research on polymer photocatalysts, composites for HAB control, and a nanobubble ozone technology that I'll spend a little more time going into some detail on next, because that's one OSU was involved in. Finally, several states impacted by HABs have supported applied HAB research and control projects within their states. I'm most familiar with Ohio's HAB Harmful Algal Bloom Research Initiative, funded through the Ohio Department of Higher Education, which has provided over $7.5 million in applied HAB research funding and is administered by the Ohio Seed Grant. New York and Florida have also specifically targeted funding towards HAB mitigation control, even more funds. There's more are more on the actual treatment though, and less on the research in general. And I expect there's many other states that are supporting this work. If anyone's aware of them, I'd be really interested to learn more, as this is something I'm very interested in. Okay, I'm going to spend the rest of my time discussing a specific project on nanobubble ozone treatment, or NBOT. That term was coined by Dr. Peter Mueller at NOAA, who has worked quite extensively on evaluating different NBAC technologies. And when we were interested in this project, he said that the one that's funded by Green Water Solutions is something that was pretty viable and was warranting for their attention. So nanobubble ozone technologies are emerging HAB control strategy that both the ITRC guidance and the harness decadal strategy identified as promising. But there's been limited efficacy data, especially at the field scale. So this project has multiple objectives with an overarching objective of determining effectiveness for cyanobacteria control and cyanotoxin destruction. It is also comparing NBOT efficacy to US EPA registered algicides, since those have been along for the longest period of time and have the best kind of track record, and also measuring the relative impact to non-target organisms. I think relative impact is important because US EPA registered algicides are out there, and we wanted to see how this compared. So you're always potentially going to have some impact, but how does it compare to what's already approved? The study is part of a larger study that also evaluated temperature effects on both algicides and NBOT treatment efficacy. And that's really important to our region and to public water systems that may have to contend with colder temperature blooms that can be more difficult to control. For example, copper-based algicides generally become less effective or even ineffective at temperatures below about 65 degrees Fahrenheit, just because the dilution is reduced there and they cannot interact with the target organism. Okay, the data I'll be presenting represents the work of a very large interdisciplinary team that helped coordinate. POPI is underlined, and the myriad of students, postdocs, and staff are all listed. Many people want to be involved in this project just because they thought it was cool, so they might not have received direct funding for it, but have assisted us in the field. We also have local, state, and federal collaborators, and our industry partner, as I already mentioned, is Green Water Solutions. This project is funded by U.S. Army Corps of Engineers, and we're incredibly grateful for their support for freshwater HAB control applied research. They're really doing probably the most at this point in the field to support this type of research. I should also mention that extension staff have been involved from the start, assisting with community involvement, field work, data translation, and as we move into results, more on information sharing. Okay, so as some quick background, since I know everyone's coming to this webinar with different levels of experience on HAB control, ozone has been used for many years as an effective drinking water cyanotoxin treatment strategy. The city of Toledo incorporated ozone into their conventional treatment specifically to address the cyanotoxin treatment challenges following their drinking water microsystems advisory that many of you are aware of. Potential problems, however, with ozone source water treatment, not at the drinking water stage, is natural organic matter can increase the oxygen demand and make it very difficult to dose and disperse ozone, especially within a larger water body, much more easier to treat in a drinking water treatment setting. Now, using nanobubbles as an ozone delivery mechanism has some distinct advantages and can potentially help overcome some of these issues. Ozone will diffuse into the water column more slowly, increasing contact times, and the Brownian motion of nanoparticles can help improve the transport and dispersal of ozone within a larger water body. Our project is evaluating the effectiveness of a new novel ozone nanobubble technology for HAB control at the lab, mesocosm, and lake scale. First, at the lab scale, we used a nanosite to measure nanobubbles being produced by the NBOTs. We confirmed nanobubble production, yay! And the size distribution and densities we measured were higher than some previously published values. So this is kind of just the first initial work. However, we also noticed some visible production of bubbles, and nanobubbles are not visible to the naked eye. So these macro and micro bubbles left solutions so fast that they cannot be measured using the nanosite. So we used a rise-weight equation to help determine the average bubble size. Now we plan to use real-time particle size analyzers that we just acquired to better quantify and determine the bubble size and density over time in the future. Of course, ideally, we want to optimize the technology to create nanobubbles, since that is the better delivery mechanism for ozone. And bubbles. We use the indigo decoloration by ozone method to quantitate aqueous ozone concentrations and the TPG method to quantitate hydroxyl radical production. One of our preliminary findings was that the hydroxyl radical generation by bubble collapse alone is minimal and only occurs at low pH. Now, this was really important because some companies are producing and marketing oxygen nanobubble generators as a viable HAB treatment technology. But our data shows that with minimal hydroxyl radical production, treatment potential, especially to an active harmful algal bloom, is pretty limited. Now, in the presence of ozone, hydroxyl radical formation is greatly increased. But we expect this primarily result from ozone reactions and not by bubble collapse. We also found that in our smaller lab scale units, ozone and hydroxyl radical production is greatest within the units. So the TPT or the indigo is added at an influent and then measured at the outfluent or measured just directly coming out of the nozzle. That's the difference between influent and effluent. Now we're working with our industry partner to optimize transfer of ozone again to within the nanobubbles versus the aqueous phase to better take advantage of those beneficial nanobubble ozone transfer mechanisms. We also need to further assess transfer in the larger field scale units because there were some differences between scaling down to the lab versus what is being used out in the field. Moving on to some of our field trials, in the summer of 2021, we completed our first full-scale NBOT treatment trial at a small recreational lake in Ohio with a history of microsystems producing blooms. NBOT units were placed at sites E and G on the map on the right center panel, and we sampled at 7 sites within the lake. The sites circled in red. We continued to sample last summer in 2022 after our trial had ended in 2021, just as a no treatment control. Now, we originally were set to begin this trial in August, but in early season June HAB prompted local stakeholders to request that we move up our trial initiation date from August to July, and we were able to accommodate that request. We wanted to make sure a bloom was present before initiating treatment, and boy, was it present. We sampled for a variety of water quality parameters at the site in our 2022 trial locations using integrated water column samplers. I'll only have time today to go over a very limited subset of our results, but hopefully I'll give you a picture of what we found. I did want to quickly point out that this was an unusually wet summer with multiple 1 to 2 inch rain events and 1 4.5 inch rain event. These events did bring additional slugs of nutrients into the system and potentially flush nanobubbles out of the system, which did complicate our data interpretation with this trial. A little bit about the treatment itself. The upper image here shows that the north NBOT unit inlet hose on the left, 2 outlet hoses on the right. The units have a pretty small footprint and are easily moved. One case contains an oxygen concentrator, ozone generator, and controls. The other houses the pumps, and ozone is added via Venturi system. Water is then discharged from the unit using a patented nozzle designed to create the nanobubbles. And the units have automatic shutoffs if ozone concentrations are elevated or pressures drop, and alerts are automatically sent to the team members based on predefined targets. This is important from a health and safety aspect, because if you would have a ozone leak somewhere in the system into the air, that could be a human health concern. We, I think, had that happen once, but the shutoffs worked great and we were able to fix the system. Didn't have any problems in our trial this last year. Okay, this was the first long-term field deployment for these units and first time they were hooked into 220 amp service versus using generators for more shorter term trials. We did encounter some operational challenges, but worked with Green Water Solutions on upgrades to the units for future trials that I'll go into later. And it's been a great, but also iterative process with data informing design changes as we move forward. Initial ozone residual measurements were all done very quickly in the field with samples analyzed on site immediately after sample collection, literally running from the sample collection site to the spectrophotometer. After analysis, samples were held and then reanalyzed at set time points over up to over a week. We were able to observe an increase in ozone concentration over time. Now, this suggests continued ozone diffusion from nanobubbles into solution, something that had not been previously demonstrated. We are writing methods paper to help others evaluating these technologies. There's some issues with pH and the indigo method, but we'd be happy to talk to anyone interested in doing this type of evaluation. In terms of water quality, we observed an increase in cyanobacteria in June. We installed NBOT unit in July, and then we're hit by our first major rain event, including some slugs of nutrients. Concentrations increased, but then seemed to stabilize through the remainder of the month. Now, this was a trial we kind of went into blind. We didn't have a very good handle on what a safe, effective dose was. But in early August, we weren't seeing any effects on targets. We decided to add a third NBOT unit and double the ozone concentration from 30 to 60 grams per hour at each unit, always making sure that our residual was low enough to not cause an impact to targets based on available literature. Now, cyanobacteria concentrations at that point decreased and remained low at all sites for the remainder of August, even after another 2.5-inch rain event. We also noticed visible degradation of cyanobacteria at the treatment site in July and a shift to diatom dominance. Diatoms are the good guys at the treatment site in August. Cyanobacteria were still present in our farthest downstream dams at this time, however. Now, our molecular data showed a genera shift, which was kind of interesting, from Aphanizomenon and Rhiphidiopsis to Planktothrix at all our sites following increased ozone dose. We don't have the field community analysis yet to show, shift to potential diatom, but it was interesting that the ozone also seemed to impact which genera were dominant on the cyanobacteria side. Microcystins in the trial never exceeded the recreational health advisory levels. And the Lake Association we are partnering with said this was the first summer in recent memory where scums did not cover the entire lake in August, and recreational microcystin advisory are not necessary. Overall, the local stakeholders thought the trial was a success. We did screen for multiple toxins at the start of the trial, and saxitoxins were also detected via ELISA and confirmed with LCMS. These emerging toxins are often not sampled for, and this was kind of a surprise for the Lake Owners Association that they were dealing with a dual toxin bloom. For us, the source of saxitoxins could be from the pelagic cyanobacteria or from the benthic cyanobacteria mats that intermittently appeared at the lake surface throughout the trial. And since control strategies are offered different for benthic versus pelagic blooms, as I mentioned earlier in the ITCRCA guidance, it allows you to select for those differences. The source of saxitoxins is of interest to both us and the local stakeholders. So the UF team cultured several benthic cyanobacteria strains from these units, or from the mats, sorry. And through sequencing of the cultures, additional saxitoxin analysis, and metagenomic investigations of the pelagic samples, which are forthcoming, we hope to determine the source of the saxitoxins. To date, I'm not aware of any genomic data that has shown what the source of saxitoxins are in the state of Ohio. Anyone who has that data, very interested in it if you have it to share. We also collected some drone multispectral imagery, and on the left panel, you can see the early July bloom. This is before treatment, or maybe that was June bloom. That was, I'm sorry, or no, early July, but it was right before treatment was initiated. The decreased bloom severity in August is shown in the central panel, and a bloom resurgence after treatment was discontinued is shown in the panel on the right. Now, we think this late season bloom was exacerbated by the Lake Association restarting a subsurface aeration system that they had after the NBOT trial had concluded. They had subsurface aeration installed as a HAB control mechanism that was not working. I mean, potentially reduced the bloom severity a little bit, but it was, obviously they're still experiencing harmful algal blooms. But they were kind of desperate. They saw the bloom in October. We had discontinued our trial, or they started to see some cyanobacteria resurgence. And we think what happened was that may have resulted in some release of phosphorus from sediments. Now, I put that in there as an aside, but it's really kind of a caution that sometimes no treatment is better than treatment. So, be wary of what a change in your system might be. Okay, moving on. Without a control, it's really difficult to predict how the sylvan bloom would have progressed without NBOT treatment. So to place our result in context, we plotted the cyanobacteria concentrations over time at our neighboring Grand Lake St. Mary's, which usually has a Planktothrix-dominated bloom, but once every 10 years, it seems to switch to an atypical Aphanizomenon-dominated mixed bloom. And in this case, this year, the Aphanizomenon bloom was very similar in makeup to what we were seeing at Sylvan. Now, while the Grand Lake St. Mary's and Sylvan blooms trended similarly in June, the Grand Lake St. Mary's bloom increased more dramatically in July after the start of the Sylvan treatment. But of probably most interest is when the ozone dose was increased at Sylvan in August, and the cyanobacteria concentrations at that point declined by more than half. At the same time point, concentrations at the Grand Lake St. Mary's increased by 50 grams per liter. So Grand Lake St. Mary's also experienced fish kills and issued a no contact recreational advisory due to microcystins detections above 50 micrograms per liter. So again, potential evidence that the treatment was having a positive effect at Sylvan. We also observed higher cyanobacteria concentrations in 2022 as depicted by the black squares as compared to our treatment year, which were the colored circles. I don't have much time left, but wanted to at least provide some preliminary results from our trial this summer at Grand Lake St. Mary's West Beach. It had at this point transitioned to a Plankothrix bloom again. This lake is really the poster child for freshwater HABs in the United States, and typically has a year-round persistent million cell per mil Plankothrix bloom that result in lake-wide microcystin concentrations that have resulted in over 100 up to 150 micrograms per liter. The bloom also is pretty consistent across the entire lake. Other control strategies, including aeration, phosphorus binding agents, and in this case, Foslock, and registered algicides, been trialed at this little kind of enclosed treatment area that I have depicted on the inlet aerial photo. And they've been tried at this location with little to no effect. In the case of algicides, which typically have the most success at HAP control, cyanobacteria reduced following application, but quickly rebounded within a week or two. Now, for our trial, we installed 4 NBOT units at the semi-enclosed State Park Beach. The State Park later installed a plastic curtain barrier after treatment started in June in an attempt to limit water transfer between the untreated main lake and the beach treatment area. Now, they had a previous curtain installed during the other treatments, but it had failed. And so they had a new curtain installed, but unfortunately it didn't arrive until after the start of our trial. 2 60-gram-per-hour units with 4 total outlets were located on the east side of the beach enclosure on the upper left panel, and 2 units on the west of the beach. Some pictures of us working on the beach, sampling kind of between the 2 units. This graph shows the composite beach sampling microcystin data for our sites in blue and 2 neighboring beaches in orange and red. After the curtain was installed in June, we started to observe a decrease in microcystins at the treatment location relative to those neighboring beaches. The treatment beach was the only beach that remained open throughout August and during the Labor Day holiday weekend, which was seen as a success by the state park managers. Now, sampling at our adjacent sites, however, showed that the treatment zone was very limited, mainly to just kind of that beach zone. And microcystin concentrations increased the farther from the units you moved. So, of course, we were hoping to treat that entire enclosed area, but really just had more of a limited impact. But it was at the area of most concern to the beach managers. A little bit on some of our other data we collected that didn't include that before. Based on organic matter SUVA characterization, natural organic matter at the outlet sites shown in red was generally more aliphatic compared to the inlet sites shown in yellow. And this is just another indication of oxidation treatment occurring at those outlets. Okay, finally, I can share some data from our recent mesocosm trial. We compared copper and peroxide-based algicide treatments to both NBOT treatments, at 2 different doses on a Lake Erie mixed Microcystis indolix sperm and bloom. It wasn't a very severe bloom because we really were also interested in the impact of the non-targets that were present in this mixed assemblage. One of the goals of this trial was to determine relative HAP treatment efficacy and, again, the impacts to non-target organisms. Our low dose was less than 1 milligram per liter, and high was approximately 4.6 milligram per liter. And as in prior experiments, we saw increases in ozone concentration and held samples over time. Oh, no. Okay. Sorry, I took a second to load that massive slide. Okay, we'll have to go back to. Oh. Okay, there. Sorry, I clicked too quickly. In general, water clarity improved in the NBOT tanks 48 hours after treatment at both doses. The control tanks are located in the left panel, and the low NBOT dose is the middle panel, high NBOT dose is the right panel, as compared to the controls. And I will say one of our controls did have kind of a unintended bloom crash, the one at the bottom, which is again why you do replicates, is you want to make sure you have a good control. We're not exactly sure why that happened in that one tank. Water clarity was decreased in the copper algicide tanks. Those are the middle panels. Again, the control panels are on the left, but improved slightly in the peroxide tanks, which are to the right. Dissolved oxygen increased in the NBOT tanks. Initially, that's the RDO, the blue and Yellow lines on the upper left panel. While DOM or dissolved organic matter increased in the copper and peroxide tanks in the upper right panel, where we saw a decrease. So again, indicating oxidative treatment at the NBOT site. DOC increased in the peroxide tank. DOM was more aliphatic in the NBOT and peroxide treatment tanks, consistent with, again, potential for oxidation of organic matter from those treatments as compared to controls and the copper treatments. Now, we're still analyzing the toxin data and conducting cell counts for this trial, but wanted to provide some preliminary information. So to summarize at least this project, NBOT units produce high densities of nanobubbles. Yay, that's what we were hoping for. Hydroxyl radical formation by collapsed nanobubbles alone is minimal. This is critical when you're looking at non-ozone nanobubble treatment. Ozone concentrations in held lab and field samples increased over time, demonstrating release of ozone from nanobubbles. At our Sylvan trial, we had severe precipitation events, inconsistent ozone at our initiation. It was a new trial, so evaluation of efficacy of treatment was pretty complex. However, recreational advisories were not necessary during the trial, and local stakeholders considered it a success. At the Grand Lake St. Mary's Beach trial, microcystin concentrations at the treatment beach were lower than control locations in neighboring beaches. Advisories were reduced at the treatment beach. However, the treatment zone was limited, so definitely need to do a little bit more on adequate dose, especially for these really severe cyanobacteria blooms. In our mesocosm trial, water clarity increased in the NBOT tanks and DOM decreased in the high dose tank, which was seen as beneficial. And I didn't provide this information yet because we don't have it quantitated yet, but I'm just looking at some of the preliminary zooplankton results in the phytoplankton community analysis. Our folks that were looking at the sample said, basically everything's dead in the high dose copper tanks, and most things are dead in the hydrogen peroxide tank, whereas the NBOT tanks, the zooplankton community, look very similar to what it did in the control. So just have some anecdotal data to share, but we're expecting that the NBOT treatment did not have an impact to non-targets, which is again critical for when we're looking at permitting and a better understanding of impacts of this new innovative technology. With that, I will take some questions. I think I'm right at 30 minutes, so be happy to answer anything that's been entered in the Q&A panel.
We have lots of tasty questions.
Woohoo!
So that said, Heather, if we don't get to them all, do you have a few minutes to type answers after we move on to the next presentation?
Sure. I will stick around at least through lunch for sure.
Great. So let me get to our first. Well, let me first bring up this again.
Oh, please no.
Oh, well,
that's okay.
Your choice, really. So the first question from Ed Weinberg. O3 plus nanobubble technology combined indicates an expensive treatment process. Any efforts to reduce O3 generation costs?
So I will say that the treatment, we are looking at costs, and I don't have all the cost data yet, and that's something we are hoping to provide when we summarize all of our data. There are some electricity costs for sure. But I will say that the state park was looking at the electricity costs for the treatment and said that it was manageable, and they were actually interested in purchasing units after this trial because they said compared to some of the other control strategies that were ineffective, this one was at least working the best. So while there are costs associated with this, I think in some cases, especially if you're dealing with severe cyanobacteria blooms, it may be cost efficient to use this technology.
Great, thanks. Robert Ripstra asks, do harmful algal blooms impact the oxygen level in shallow inland lakes that could cause a fish die-off?
Oh yeah, this question should go to Eugene. He's my fish guy. But yes, so when you have a harmful algal bloom, you can have actually super oxygenated water and then crashes to no oxygenated water following the kind of the natural life cycle of algae as they consume and release oxygen. So a lot of times you'll see fish kills in the morning following a harmful algal bloom because you've had that cycle occurring over the nighttime. So yes, you can have that. So there is an added benefit of ozone technology, in that you might have some additional aeration benefits, as I saw in that one case. Because in the mesocosm tanks, we weren't running the tanks continuously. In that trial, we literally just put the outlet nozzle into the mesocosm for a period of minutes and then stopped treatment. So that's why you saw kind of the raise in oxygen and the decrease. But in an actual site, you'd probably be using continuously adding oxygen, so that could be an added benefit similar to aeration or other technologies that would have that benefit for the fish.
Sure. I mean, that's of course, green things are net producers of oxygen when the sun is shining on them, and consumers when the sun goes down.
Better said, Eugene. So I have extension folks involved in this project.
Oops, and there was a question that was upvoted in the interim, and I just dismissed that as though it was the question we were addressing now. So I'm not sure what question I just dismissed, but if you typed a question. Oh, you know what? I think I remember it. It was from Fred Milan, and he was asking if there is sufficient, if there are renewable energy sources available that could power NBOT technology, things like solar driven systems.
Unlike your traditional, kind of aeration systems or your solar bees, the energy requirements are pretty big. So I'm not sure. I mean, you could probably put a larger solar array, or something up there that, I won't say it's impossible, but it is a little more challenging than some of your your smaller aeration type units. Again, it's because of the needs of the ozone generation, the contractor. Now, and the pumps, of course, to kind of, you pump water in and you pump water out. So there are other folks that are looking at ways to, how we can optimize the system and make it, as least power intensive as possible. But yes, I won't say no, but it would be a challenge.
Even I think conventional aeration systems, solar driven systems still tend to be underpowered, and battery storage is not good yet. It's improving, but it's not there. John Gatto asks, any thoughts on scalability of NBOT for real world HAB control?
So Sylvan Lake was a pretty decent size in the lake, and we only had a couple small units there, and seemed to have an impact. Again, I think when we're looking at getting a better handle on dose, how severe the bloom is going to factor in, as well as the size of the lake. Our partner is working on developing kind of larger units that are more in a trailer that can be sequenced. There are other companies that have larger units that are being deployed. So yes, this is potentially scalable. It's just going to take a little more power and more effort, but it could be scaled.
Great. Ed Weinberg is a prolific typer of questions for this presentation. I'm glad he found it interesting. Mesocosm tank tests, any fish in the tanks, any aquatic toxicity data for NBOT treatment?
So we were mainly looking at zooplankton. There may have been some fish fry in there. It's basically just the mesocosms are set up to pull in water from Lake Erie. So anything that was in there, but I don't know. There is some, as anyone who's done university research knows, as soon as you involve vertebrates, you have to do a lot of other legal hoops to jump through to get kind of approved for those plans. So we're not specifically looking at vertebrates. I do think that more controlled lab-based toxicity experiments on model organisms is important and should be conducted. This was kind of our just first foray into impact to non-targets, and at least on as far as the zooplankton were concerned, we have some preliminary, some promising data, especially compared to copper, because I will say copper, with the doses we use for copper and peroxide were the max permissible label doses, so they were higher doses of copper, but they were label permissible doses. So we did see some pretty high impacts in some of those other tanks.
Great, thanks. Gina LaLiberte asks, did the ozone nanobubbles affect non-target organisms? I've seen some presentations in which it's clear that this technology kills almost everything, even beneficial algae like diatoms.
Yeah, so dose is the issue. So as in anything, you take enough aspirin, it'll kill you. So I think that's why when we're looking at this, we're looking at the ozone residual and maintaining a lower ozone residual. There has been some data on just ozone treatment in general on what residual is considered acceptable for impacts for non-targets. So we're always trying to maintain below that. So that is a question. Now, what's interesting though is, since we're encapsulating the ozone within the nanobubbles, that is changing the delivery mechanism too. So there are some things that I think still need to be teased out, and potentially through more lab-based toxicity studies, we could have some better answers. Because it's not like we're just injecting ozone in the aqueous phase, which might have a larger impact than within these nanobubbles.
Great. Val Beasley asks, what, if any, risks exist with regard to inhalation of ozone by workers or other nearby air-breathing organisms?
Yes, so this is why I said I would not work with this technology unless there were alarms and automatic shutoffs, because yes, you can have a leak and that could be a concern. As I said, I think we had one very small minor leak, never even enough to kind of smell ozone, but it was enough to trigger the alarm to turn off. We set a pretty low threshold there. So yes, if you had a major leak, if the whole hose popped off, you could release ozone. Now, since these treatment technologies, I will say, are taking place outside, not in an enclosed area, all of the units are out there, the risk is less, but it was still something that we were concerned about and made sure we had adequate controls in place to prevent that type of mishap.
Indeed. Ellen asks, is there potential to use NBOT as a preventative measure for places in which HABs are frequently found?
Yeah, that's another question. I think this might be the most efficient use of NBOTs is if you can put in, lower controlled doses at the beginning of a less severe bloom, then that might be more beneficial. We have not investigated that yet, but I think there potentially is some room for that. Of course, then you have to weigh the cost balance sheet and the benefits, because it is a little more a little more expensive. But I think in each case for our full scale trials, we went in and the bloom was already really severe. I would like it, with additional funding, additional time, it would be good to go in when a bloom is just initiating and is lower cells per mil to see if we can inhibit that and be more effective then. Because I know even for algicides, we've always recommended, go as soon as you start to see that shift in dominance from the beneficial algae to cyanobacteria, and not wait till you have a really severe bloom before you treat, because it's even for algicides, which are generally pretty effective, is much harder to treat when you have an active bloom. So, future research for sure.
Right.
Sorry, long answer.
It's a good answer, right? That's an appropriate answer. Ed asks, for the test unit shown, gallons per minute feed rate, what was the total surface area influenced?
Ooh. Oh, each child's a little different. It'd probably be best if he emails me and I can provide those details. I don't know everything off the top of my head. I know the current units are kind of rated at 55 gallons per minute per unit. So that's kind of what we're treating. I know for our Grand Lake St. Mary's enclosure, we estimated with perfect water transference and no interaction from the greater lake outside of that curtain with the inner lake, it would take 30 days to move the entire volume of water within that area through those 4 units. So maybe that gives you a little bit of an estimate, but I'd be happy to follow up with additional details. Great.
Well, if Anne doesn't hear this and get your email address pasted into the chat, I will do so during the next talk. Give me a moment to scroll back up to the top of my question list. Bree Olson asks, how many locations did you have the NBOTs? Are they a circulating system taking lake water from the lake and adding ozone by a pump back in?
So I think I may have, sorry, again, was really quick on that description, but yes, they are a system where you kind of locate inlet pumps. It's far kind of away from your outlet pumps, so you don't have a closed circuit as possible. And it is pulling water in at that 55 gallons per minute and then putting it out. You pull in one larger diameter hose, and you have 2 smaller diameter outlet hoses to kind of increase the pressure differential there.
We're getting close to the end of our time. We might only have time for 1 or 2 more questions. And again, if you have just a minute to stay afterwards and type answers to the remaining questions, it's appreciated, of course. Bryce Hoppy asks, could you speak to the spatial spreading and reach of the treatments? To what degree, if any, is wind-driven mixing of this on this particular treatment?
So one thing that's interesting, and we've debated doing some tracer experiments or things to try and to better quantify that. But nanobubbles themselves are kind of unique in that Because of they move via Brownian motion, when they're dispersed, the charges repel each other and they move pretty quickly up and down in the water column. How efficiently they're moving up and down the water column and dispersing, hasn't, we haven't calculated that. But in general, they're not moving up to the surface. We usually put the outlet nozzles, a foot or two underneath the water surface and kind of spread it out so that way they could get that biggest spread and impact. But unfortunately, we don't have good hard numbers on that. But ideally, based on the theoretics of how the nanobubbles work, we should get a pretty good dispersal of those nanobubbles throughout a lake. And I know that in a flowing system, so if you have ideally you might locate units at the inlets to a reservoir. We have kind of water flowing into the reservoir, which would help kind of aid in that transference and dispersal. For our Sylvan Lake, we basically went with where power was located because we weren't going to pay for additional installation of power where they had their aeration units already. So that wasn't necessarily the most ideal location of where we placed the units, but it seemed to work okay.
Great. Thanks so much for all your time, Heather. It's been a great session. I really was engaged with the questions. Please begin typing your answers now. For those of you who are interested, note that Anne has pasted Heather's email address into the chat. So thank you so much for your time, Heather.
Great. Thanks to you, Eugene. It was a pleasure being here.
Thank you. So I will now take the moment to introduce Leif Olmanson, a research scientist at the University of Minnesota with more than 25 years of experience developing remote sensing applications to create temporally and spatially rigorous data sets of water and land resources for large area ecosystem characterization. It's particularly interested in developing field validated image processing methods implemented in automated geospatial analysis systems to gain better understanding of natural environment. I'm eager to hear his presentation, so feel free to read a little more detail on your screen as you find appropriate. And without further ado, let's hear from Leif.
Okay,
Thank you for being here today.
Sure. Let me see. Let me share my screen first, and then I'll get the presentation started. So you can see that, right?
Looks good.
Perfect. Okay, cool. Move this over here. Okay, so let's see. Let's go back one. Okay. Yeah, so I'm going to talk about the work I've been doing using satellite remote sensing for water quality assessments, and specifically how we've automated the image processing procedures with the help of David Porter from the Minnesota Supercomputing Institute. And then I'll talk about how we can use the data that we're producing to identify lakes that are prone to cyanobacteria blooms. So in Minnesota, we have a really good lake monitoring program. So we have lots of field measurements collected on our lakes. At the same time, we have satellites that are regularly collecting imagery. So what we do is we find some good, clear late summer imagery when algal blooms are at their maximum, link that up with the field measurements within a few days of the image, build a statistical model of that relationship, take that model, apply it to all the lakes on an image. We do that for enough image, we come up with the water clarity for the whole state. So what we are using these procedures, we have 9 statewide assessments since 1975. 10,000 or more lakes, over 10,000 lakes are included in each of these assessments. These are around 5-year intervals due to the availability of the imagery back then. When we started this with one satellite every 16 days, so clear imagery was fairly limited. So we're basically, and then it takes a lot of time to process them. So it's every 5 years seemed like a good time frame to use. We use it for analysis of spatial and temporal trends and causative factors. And with that, we have a publication, and we're currently updating that analysis. But the time keeps moving, so things keep moving too. So trying to get ahead of that one. One unique thing that we did back in 2002 is we made all of our data available in a lake browser. So it's a Minnesota lake browser. It's an online resource, Google Earth format type thing, which I'll show you a little bit. But it has been very popular with thousands of unique visitors monthly. And it's been recently updated with all the new data that we've created with our automated system. So we have a lot more data in it than we've ever had in the past. So kind of a paradigm shift there. So we have all these new sensors, and there's a lot of new sensors coming up. So we have higher spatial, spectral, radiometric, and also temporal resolution. So not only can we map water clarity, we can also map the factors that control water clarity, things like phytoplankton, suspended solids, and dissolved organic color. So the spectral characteristics of the sensor really determine how well it can measure different water quality variables. And so I have some spectra down here from some lakes in Minnesota, showing kind of the broad range that we have in Minnesota. And then I have the band placements down below. So if we have just water for a typical Minnesota Clearwater Lake in Minnesota, we're gonna have a peak around 550. If you have like a mine pit or something where there's really nothing in the water, you're going to have a peak over, or ocean water will have a peak, at lower wavelengths than this. When you get chlorophyll in the water, you're going to have this important band here, this peak at 705 due to the particles scattering. And then you have absorption due to photosynthesis at about 670. And then there's another absorption trough at around 620. And that's due to phycocyanin. So if you really want to get phycocyanin, then you need a band in this area. Unfortunately, that's kind of limited to the size of lakes you can do. For CDOM, the brown stuff in the water, basically color dissolved organic matter. It absorbs very strongly in the shorter wavelengths, and that decreases as the wavelengths get longer. And so you can see how that affects the spectra. So if we have just CDOM, you're going to have very little signal at all coming out of the lake. But once you have some algae in there, like this yellow one, you'll see how it pulls the shorter, compared to the green one, you can see how it pulls the shorter wavelengths down, leaving the longer wavelengths intact, so it can still be used for chlorophyll measurements. Now, if we look at the band placements, we can see the Landsat. That's been the workhorse for the last 45 years or so. That's satellite's been around forever. It's typically been every 16 days. Once in a while, there'll be two of them operating at the same time. But for the most part, it's been every 16 days. So it's kind of limited. The band does not have that red edge. So it's an important band. So Landsat works great for water clarity and also for color dissolved organic matter. If we're going to get chlorophyll, we're better to have the Sentinel-2 satellite. This is more a recent satellite. So Landsat's been around since the early 70s, whereas Sentinel-2 was launched in 2015, but really only operational in the United States in the late 2017, or fully operational. So we have that red edge band is very important. And if we really want to get the phycocyanin, you need a better band set than that, and that would be the Sentinel-3, which has 300 meter data. So now we're going from way over 10,000 lakes to maybe 900 lakes in Minnesota. So big difference what you're monitoring. So really, ideally we'll use this. The Sentinel-2 will be able to get to study all lakes, and I think that's, for us in Minnesota, that's more important. For in the future, Landsat Next is coming up. A lot of scientists got together, and we kind of had a wish list of spectral bands and characteristics for a satellite. And most of our wishes were actually granted. And so with the Landsat Next will be a really game changer for remote sensing of water quality. And that's probably 2030, I think, is the latest for when it will be launched. So it's not really in the near future. So really, we got to work with what we have. And so that's what we've been doing. And a big part of that is having the Sentinel-2, which really increased the temporal coverage, which is huge, and made it really, reasonable to start doing automated image processing. Another thing with the Landsat sensor, now we have two Landsats. Landsat 9 was launched, and last year was the first summer that we had imagery available. But now we have 2 of them, so now it's every 8 days, so pretty good coverage, and big advantage with the or potential advantage for some things would be the thermal bands. So you can actually get surface water temperature, which is an important factor for harmful algal bloom development and such. So let's just look at some of the imagery that's available. This is Sentinel-2 for August of 2022. And first thing you'll notice is there's lots of cloud cover, and it's very difficult to work with imagery, lots of cloud cover. So having our automated processing techniques make that a feasible project. For all of the previous assessments, this is the type of imagery that I would have used, a nice clear image through a lot of lakes. You can study a lot of lakes at once, but that is a fairly rare occasion. If we look at Landsat, now this is with both Landsat 8 and 9, you'll see it's actually pretty good coverage, but if you take half that imagery away, it was pretty, more limited than the Sentinel-2. So now with both the Sentinel-2 starting in late 2017 and this, or full operation in 2017, and then the Landsat 8 and 9, we actually started to have something here for imagery available for processing. So we have the opportunity for near real-time water quality monitoring. So we built a system that's capable of that. So we have data coming in from the sensors and downloaded to the ground stations. From there, we can get machine-to-machine access into the Minnesota Supercomputing Institute. From there, we kind of do our remote sensing magic, things like atmospheric correction and different masks. So atmospheric correction, basically you take what the satellite sees and you correct it by removing the atmospheric effects to what you would expect on the ground. There's lots of different atmospheric corrections out there, and some work better than others, some work terribly. And it's really important what atmospheric correction you use. And so we tested 8 of them, and we found a couple that worked well for some sensors. We found the main atmospheric correction for Sentinel-2 and for Landsat works great. We can harmonize the sensor, and that's what we've been using in Minnesota. For cloud masking, we start with FMAS 4, which is a basic cloud mask. It's available, and then we take that and do a buffering around it, because there's lots of artifacts left over from that around the edges, and then there's lots of other things that are missed from that, so we have to create additional masks. But once we have that, we started processing our imagery, and right away you started seeing some anomalies. We started looking into that and found wildfire smoke was a big problem. It wasn't picked up, it was picked up in the terrestrial areas, but not over the water, so it really didn't work with what we needed. And so we needed to take care of that as well. So wildfire smoke, if anyone's noticed, it's becoming a problem in Minnesota and throughout the country, actually. So that's something you need to deal with. So we came up with an additional mask for that. So now we have just clear pixels left. From that, we can make our models. So the way we were doing models is we're basically taking all available in situ data, and we're linking it up to all available clear image assessments. And that's basically, we have the location of the samples, and we have a buffer of a few pixels around it to come up with a sample, and we link that up. And then we were able to build some models. This is, we have over 20,000 within 1 day for SecuDisc readings. And this is only through 2022, so there are 20. So we actually have a lot more data for 2021, 2022. And we're actually expanding to other areas as well. So Wisconsin and Michigan, and then throughout the country, we're going to start to gather some other data. So we'll have a larger data set to really do some good machine learning stuff. And we found we have good models now, but we found we can improve them with machine learning. And so that's kind of the way we're moving. And with machine learning, you really need a lot of training data. So that's kind of the key for that. That was like water clarity, and this is chlorophyll. For chlorophyll, it works really well, except for the very low chlorophyll levels, and so that will basically overestimate chlorophyll measurements, and so that could be problematic if you're interested in those really low. For us, I guess we're more interested in the ones that are really having these big algal blooms. So and for this group probably as well. So they actually work really well. But machine learning actually works good throughout the whole range. So that's that'll help a lot with that one as well. And we also have a lot of measurements for chlorophyll. So once we have these models, we can apply them to the imagery. And then we can provide data and maps and provide it in our lake browser. And I'll show you that in a little bit here. So we have the products we have is chlorophyll, clarity, and CDOM right now. And we have basically at lake level, we've basically take anytime we have clear imagery, we actually collect a sample and we basically put it in a CSV file for use in the database. And then for monthly data, we actually go to the monthly mosaics. So we found, so one image would be in one part of the state and others. And so by doing a monthly mosaic, we're actually able to get coverage for the whole state. So here's an example of some of the imagery. This is the Lake of the Woods Rainy River Basin up on the Canadian border, Minnesota with Canada, up in Ontario there. And basically, I'm just going to show there's lots of pristine lakes. Lake of the Woods is a very exciting lake with lots of cyanobacteria blooms, and then upper and lower red can have some problems too. So, but for the most part, the rest of the lakes up there, there's way over 10,000 lakes up in this area, and they are, fairly pristine. So I'm just going to look at. There we go. Okay, nope, I need to get it going here. Okay, so if we look at this one spot, I think I did that wrong here. There we go. So that red spot kind of pops out. So you kind of got a needle in the haystack there. So we're looking at sensing 10,000 lakes. We can actually see that. If we look at the imagery closer, we can actually see, yes, Laurie Lake actually did have a big bloom that day. If we go from our dynamic area, this is the Twin City metropolitan area. You can see the developed area and then a lot of agriculture around it. And let's go to this one. This is a good illustration of early 2017 of the availability of imagery. Due to the ramp up phase of the sensor, there wasn't really a lot of imagery over the United States, and so this is what was available for that day or that month, and this will improve as we get to the later summer here. So start this 2017 through 2020, May through October, and things we'll notice is every late summer you start to get a lot of blooms, and there's same pattern every year. More intense some years than others, but pretty much the same lakes are blooming on a regular basis every year. And so we have this record of what's happening to these lakes with this. So, and we have this type of data available for chlorophyll, clarity, and CDOM. So let's go to the daily level. So this is the daily data for daily measurements at the lake level. We have over 3 million measurements if you include chlorophyll, clarity, and CDOM. So here's this is from 2017 to 2022. So we'll look at a few lakes here. This is Lake Francis, mesotrophic lake, Tatanka, Washington, Cicada, and German, which are more eutrophic, and then Elysian, which is hypereutrophic. If we take that do the same thing for a number of years, you can see there's the Francis, Tatanka, Washington, Cicada, German, and Elysian, and we can see quite a bit of difference, but from one year to another. Elysian's really high for these 2019 through 2021, but lower in the earlier and later year. So there's a lot of dynamics, and then you have upper Cicada it's actually blooming more in 2022. Some things, different conditions each of these years. So it's really good to have a long-term record of all this data so you can see what's going on a regular basis. If we do this got to go back now. Okay, let's see. Click the wrong spot. So here's the same lakes, but just for 1 year, and just kind of seeing how it is, where these lakes are, and how different they are from one another in the same area. There's quite a variety in how the seasonal components treat them. So if we look at spatial and temporal trends, what we see is yeah, we used our previous assessments and then we used our later stuff. And we have distributions here. So we basically calculated the late summer average. So basically the dog days of summer, July through September, or mid-July through mid-September. And then calculated that through with the automated system. So we have the long-term trends. Plus we have, all these maps for each of the time steps, and found that basically northern lakes and forests, best water clarity of the state, north central hardwood forest, more mixed, and then more eutrophic in the western and southern agricultural areas. So if we look at long-term trends, we have a higher percentage of decreasing water clarity in the agricultural areas, and then we have a higher percentage of increasing water clarity in the northern lakes and forests and the north central hardwood forest ecoregion. Some hot spots of changes, big agricultural changes in this area, going from small grain to corn and soybeans. In this area, plus some hydrologic changes. Up in this area, big concern for us was all these blooms up in northern Minnesota, probably due to temperature. And you can see how, there's been actually a pretty big increase in temperature over this period. And northern Minnesota is changing faster, warming faster than any other part of the state. And any part of the United States as well. So that's a concern. Big improvements actually in the metro area. So areas that are being developed with the best management practices seem to be working. There's probably also a factor of zebra mussels. When I looked at the assessment, the number of trends, about 3% of them were due to zebra mussels, and so not all of them, but that was one of the factors. So we have 20% of our lakes had changes of 10 TSI units over this time, when that would be equivalent to a doubling or halving of algal biomass. That was about 14% had improving water clarity trends, and 6% had decreasing water clarity trends. So we're moving in the right directions for a lot of these lakes. If we look at a smaller trend, 5 to 10 TSI units, it would be 18% improving and about 9% decreasing. So we're kind of going in the right direction, but there's still work to be done, and it's definitely in certain areas. So we can look at the distribution, different ecoregions. When you got like lakes, wetlands, and forests, you're gonna have better water clarity than if you have the mixed agriculture. It'd be more of a mixed bag. And if it's just agriculture, it's going to be more on the lower end. Let's go to our lake browser. We're going to zoom into here. There's going to zoom into a certain area. So here we have, we can pick what we want to look at. We have it either by the lake, or we can do it by pixel level. I'm going to pick up chlorophyll pixel level data. And then we can do it by different months here. So this is May. We already have some blooms starting in a few lakes, but not too many. June, we have a few more blooms. July, some cloudy imagery, so maybe not quite as many imagery, but still more blooms going on. August, a lot more blooms. In September, more blooms, and then even holding out in through October. So you got a kind of a bird's eye view. You have, you know what's going on. There's a big problem. There's a lot of these lakes are eutrophic and could use some help. So how does that work? It's very expensive depending on what you're doing. We see that best management practices do work in the urban areas, so maybe that's something to think about. Here we have a full report. We can click on any lake and get a full report. Here we have improving water clarity. So this is from 2007 or 1975 through 2022. We can look at the seasonal for the water clarity, tabular data for all of the different timeframes, and then also for the monthly data once we get into 2017. We have daily data as well. So we have clarity, chlorophyll, and CDOM, and we have that for every clear occurrence. And so you can see there is quite a bit of data available for that. And then we can rank this. So we can start looking at lakes in different delineations, and we can give it a ranking, how it ranks. So this statewide, this is clearer than 75 74% of the lakes. Seasonal chlorophyll, tabular data, and then we got some land cover data there. There we got that. We're also expanding to Michigan and Wisconsin. These are preliminary maps created with our Minnesota models, and we will be redeveloping our models with a lot more data. We have this is 2021 for the states, and we have lake level and pixel level data. Pixel looks includes the big lakes here, so that's why I included the pixel level. And here's 2022 for that. So we have lots of data. We can start looking at tabularly. If you look at the data, we have Minnesota has twice as many lakes as Wisconsin and Michigan for using the same criteria. Wisconsin actually has, and Wisconsin and Michigan actually have a higher percentage of clear lakes than the really clear lakes than Minnesota. So kind of interested in looking into that a little deeper. If we look at the ecoregion level, so the Northern Lakes and Forest ecoregion, since that encompasses all 3 states, similar pattern where we have higher percentage of clear lakes in Wisconsin and Michigan. So that's something to look into. We're interested in looking at other areas, some of the more dynamic systems like reservoir systems down in Georgia and Alabama, and throughout the Florida and other places to kind of get an idea of how well our models work in other areas or develop new models that'll work everywhere.
Quickly, Leaf, we're about 3 minutes out.
3 minutes for the full thing. Yeah, okay. Okay, so then the last thing I'm just gonna, with all this data, we can start to model. The one thing with the satellite imagery, if you're gonna do a harmful algal bloom monitoring or something, you're gonna not have clear imagery all the time. So you really are gonna have to model it. And so with having the data, we have the historical data, bring in some temperature, precipitation, climatic type data, thermal data from the Landsat Century lake water temperature models. You could come up with a way of actually predicting the harmful algal bloom potential. And that's kind of where we're hoping to head at some point here in the future. And with that, I have just a paradigm shift. We have all this data. How can you use it is the question. And for that, I'm done then.
Thanks for lots of interesting information. We'll probably only have time for 1 or maybe 2 questions live. As I asked Heather, do you mind staying on a bit afterwards and typing answers to as many questions as you can frenziedly type after the fact? Assuming you will. So the first question from Heather Krempa: have you explored the use of these models on flowing waters?
At the pixel level, they're on flowing waters. Flowing waters are very dynamic, so the timing is really different than static waters. So the matchups are going to be very limited for satellite imagery. Satellite comes over once a day, once a week or something. So there probably won't be that many matchups, but the models we develop for inland waters, static waters probably works just as well for that. And all those are included in the lake browser. At the pixel level, you can see a lot of the trends and stuff. And it is, like up in St. Louis River estuary or something, you have all the dynamics of the CDOM, the chlorophyll, everything is all there. So it's actually kind of interesting. Yes, definitely.
Great. So I'm going to read one more question. Do you have time, Leaf, afterwards to type some answers to the questions that are in the question and answer panel?
Sure.
Okay. So the one last question I'm going to get to, Sarah Davis asks, how does your work compare with the Cyan app developed by the EPA? And she posts a link to the Cyan app if you're not familiar. I have it downloaded, of course.
Yeah, I'm very familiar with it. I think the Cyan project, there's also the, what's the other one? Cyano Lakes is another one. Is actually works pretty good too. If you haven't seen that app, you might want to download that one as well. The thing with that's using the Sentinel-3 data, and so it's the larger lakes. And from the examples I've looked at, it doesn't always, basically like up in the St. Louis River estuary, Lake Eloise is actually, there's a lot of clay in that lake. And so it says there's a whole bunch of cyanobacteria or whatever the counts of, of that. So it's not, I don't know how accurate it is everywhere. So that's something to take into account. But this is just doing lakes. This is doing all the lakes. So we have, over 10,000 lakes, where that's probably, 500 lakes or something in Minnesota are being assessed with that. And yeah, I think I'd look at the Sino lakes too, and do some comparisons between the two of them.
Great. Thanks so much, Leif. Again, if you can take at least a little time to type some answers, there's still plenty of questions in the question and answer panel. Sorry, we couldn't get to many of them.
Yeah.
Thank you for your time today, Leif. It's
Sorry, I went over.
quite all right.
Let's see, let me stop sharing there.
So I'd like to introduce our next speaker, Shua Chen. Shua is a PhD student in the Department of Agriculture and Biological Engineering at Purdue. Shua's research interest is forecasting harmful algal blooms using machine learning and remote sensing. Her goal is to improve the rapid quantification of the biochemical state of inland waters by integrating satellite, hydrodynamic, and meteorological data using machine learning approaches. So Shua, if you're ready, I'm keen to hear your presentation. A reminder while your presentation is coming up, type your questions in the question and answer panel, all please.
Can you see my screen?
Looks good. Thank you.
Thank you. Good morning. Thank you so much for having me today. My name is Shua Chen. I'm currently a PhD student at Purdue University. The topic that I'm going to share today is remote sensing of Midwestern U.S. water quality. The presentation will provide an introduction to our ongoing project. In the Midwest, inland water like the lake and reservoirs are very important because they provide critical ecosystem services. For example, they provide water supply for residential, industrial, and agricultural users. They also provide recreation spaces and electricity generation. However, the water quality is experiencing huge success from both environmental and anthropogenic events. For example, the agricultural lands may have phosphorus and nitrogen leaching, and the precipitation and the consequently runoff will transport nutrients from upland sources to water bodies, which can cause cyanobacteria blooms. To reduce the impact on both human and ecosystem health, it's critical that we can effectively identify the harmful algal bloom in multiple inland waters. The Indiana state agencies conduct regular sampling from May to August every year. The water sample will be grabbed and analyzed in the lab for the cyanobacteria cell count and the mycotoxin. For example, if the cyanobacteria cells is greater than 100,000 cells per milliliters, they will put on advisory alerts. If algae toxin was tested in the water, they will put on caution alert or even close the beach. However, the current harmful algal bloom monitoring is very expensive and labor intensive because they need somebody to go out to visit every site to grab the water sample and analyze them in the lab. We still need tools that can quickly quantify the water quality and the harmful algal bloom potential within lakes across the Midwest. Remote sensing is promising for large-scale and real-time monitoring for harmful algal blooms. Our team has applied remote sensing on monitoring the water quality in the Midwest. In 2013, our team estimated the temperature of stream flow using the thermal infrared imagery. Then our team developed empirical model to quantify the water quality parameters in Wabash River use hyperspectral data. In 2016, our team published a spectral biogeochemical database of Midwestern rivers. Besides the rivers, we also care about the plume because the river will carry nutrient and sediment to the lake and then cause the plume. The plume can be the large plumes is easier than the narrow river to be observed by the remote sensing data. And they are very important to the water quality and the potential of harmful algal blooms. Thus, in the same year, our team developed a biogeooptical model to estimate the water quality of the river plumes in the southern Lake Michigan. The empirical model and biogeooptical model that developed in our previous research will be used for our ongoing project to focus the occurrence of harmful algal blooms. So based on our previous work, our ongoing 3-year project is to improve the rapid quantification of biogeochemical state of both river and lake water in Wabash River watershed. We have 2 goals. The first one is to quantify the link between optical, thermal, and water body properties. And the second one is to identify the contributions of the water inflow to river mixins and the formation of harmful algal bloom in both river and reservoirs. Our output will be an improved model and improved harmful algal bloom prediction model. In the coming summer, we will start our regular sampling of the biogeochemical states of both of our study areas. That will be our first-year campaign. Our proposed sites will focus on 6 reservoirs surrounding Purdue campus. They are Lake Freeman and Shaver, Mississippi Reservoir, Eagle Cray reservoirs. We are also considering 2 other reservoirs. They are Guys and Moss because they provide water supply for Indianapolis. Some water quality parameters like TSS, pH, and DO will be measured by a YSI song, but most of the water quality parameters will be analyzed in the lab using an IC and an HPLC. At the same time, we will also collect remote sensing data. On the boat, we will collect ground reference spectral reflectance by a handheld spectrometer. At the same time, we will fly UAV to collect the multispectral, hyperspectral, and thermal data. We will also make use of the images from different satellites. All this data will be used to develop our improved prediction model. The whole framework will integrate three parts. The first part is to use a biogeooptical model to predict the optical active constituents like the chlorophyll A, total suspended solids, dissolved organic carbons, and ash. The heuristic models will be used to predict the non-OACs like the nitrogen, phosphorus, and pH. And a mixing model will be used to estimate the characteristic of the river plume mixing. And all this information will be used to predict the potential occurrence of the harmful algal bloom. Firstly, the remote sensing reflectance will be input to the biogeooptical model to predict the OACs. Our team have already developed biogeooptical model in our previous research. Here is the map of concentration of non-algal particle, chlorophyll, color, dissolved organic matters. And the series and the basic theories is that the reflectance can be determined by the coefficients of absorption and back clustering. And take the absorption coefficient as an example. It was the combination of water, non-algal particle, chlorophyll A, chlorophyll, color disorganic matters. And the concentration of these components are, the coefficient of these components is relative to its concentration. So by solving these relationships, we can estimate the concentration of different OAC. The Biogeometric optical model cannot be used to retrieve the non-OAC because they don't have any effect on the spectrum. So, but however, sometimes the non-OAC may be more important for the water quality and the potential of harmful algal bloom. So we will develop heuristic model. It will make use of the OAC that stimulated from our Biogeometric optical model and some additional environmental data like precipitation, water depth, nutrient, and extra to predict the non-OAC. Also, the plumes will carry some nutrients, so it's very important to know how long the plumes will stay on the water surface. Here, we will develop a plume missing model to predict the fate of the plume at the reservoirs. The thermal imagery, wind speeds, flow rates, and pressure will be improved to a 1D water color missing model and predict the characteristic of the missing. For example, this is for example, the water temperature of the Grand Lake is higher than the Lake Michigan. So in this case, the plume formed from the Grand rivers will stay on the surface of the Lake Michigan. In the future, the instilled water that collects in fields will be used to calibrate and validate our missing model. Here is some preliminary results. This is our first year, so most of the results are based on the historical records. Among the 6 reservoirs that we propose, we think we will pay more attention in Lake Mississauga Lake because firstly, we found that harmful algal bloom occurs almost every year in Mississauga Lake. From 2014, the microsystem was tested frequently in Mississauga Lake, and we also identified the most significant harmful algal bloom in 2019. It is the human recreation advisory alert and almost hit the prohibit alert, prohibit threshold. So we will use some vegetation index to see what was happening in 2019. Vegetation indices is designed to enhance the vegetation, to capture the vegetation signatures. For example, the vegetation reflects high in the NIR bands, but low in the visible spectrum. Here is around the red band. And this is how NDVI was developed. In previous research, NDVI is frequently used to abstract the algae. For example, in general, the NDVI of water is negative, but for the harmful algal bloom is positive. So the closest available Landsat 8 image that we can obtain was on September 7, 2019. As we can see, the positive NDVI above the water are probably the harmful algal bloom. If we only the current sampling strategy is take the water sample on just single site. If we only collect the sample and test them on the single site, we may ignore the rest of the harmful algal bloom. In the future, we will make use of Landsat 9 to have a 8-day revisit time, but now because Landsat 9 was launched in 2021, so for this harmful algal bloom, we have to use Sentinel-2. As we can see, before August, the NDVI for this point is always negative. So which means that there was no harmful algal bloom. But during the August, they are always positive, which means that they may have a harmful algal bloom last for the whole August. And another interesting phenomenon is that we found that NDVI is negative on August 24. But according to the IDEM report, that should be a harmful algal bloom. So we checked the discharge of the discharge data, and we found that the discharge is pretty high that day. So we believe maybe the algae was covered by the water, so we cannot see it from the remote sensing data. Additional work that we have a comparative, including predict concentration of chlorophyll A using classical machine learning methods. We found an open source national database called ArcStats. This single database provides in-situ water quality measurements of chlorophyll A, SDD, DOC, and TSS. And they also pair these measurements with Landsat spectral reflectance. So based on this data set, we start our own exploration analysis. Firstly, we use the concentration of chlorophyll A as a proxy for harmful algal bloom because the algae use chlorophyll A to generate chemical energy to grow, and they're green, so it's very easy to be observed by the remote sensing data. And also a recent study showed that the concentration of chlorophyll A is strongly relative to the microsystem. And then we calculate candidates vegetation indices based on the spectral reflectance. We firstly uncover the relationships between concentration of chlorophyll A with these predictors using Pearson correlation coefficient. And then 4 classical machine learning models were trained. They are multilinear regression, super-vector regression, random forest, and extreme gradient boosting. The whole data set was divided into 80% training sets and 20% testing set, and R-square and mean square error was used to validate the accuracy for the test set. Here is the relationship between the concentration of chlorophyll A and vegetation indices. In the 2, 3, 4 here indicate how many bands these indices use. In this slide, we want to highlight 3 points. First one, the NIR band is very important because RGCI and 2BDA, they are both ratio indices, but 2BDA use NIR band and it has higher correlation coefficient with chlorophyll A. And we also want to highlight that the normal difference indices can be very useful because the main difference between 2BDA and NDCI is that NDCI is a normalized difference index and it has higher correlation coefficients. It may be because that the normalized difference indexes can reduce the difference in the atmospheric correction and radiometric calibration. And lastly, we've also found that using more band not always have higher correlation coefficient. For example, there are some 4-band indices, but they don't have a significant higher correlations with the concentration of chlorophyll A than the 3-band indices. If we only use the vegetation predictors to predict the concentration of chlorophyll A, the R square is about 0.21. So we thinking about maybe we can make use of the rest of the institute measurements. Here is the correlation between the concentration of chlorophyll and the other institute measurements. Overall, the TSS and DOC have higher correlation coefficient than the Sanchit. It maybe, for example, the DOC can provide energy for algae to grow, and maybe this is why it has higher correlation coefficient than the Sanchit. Also, if we only use these three parameters to predict the concentration of chlorophyll A, the R square is about 0.34. So lastly, we combine the vegetation index and the water quality measurement to predict the concentration of chlorophyll A, and we found that the concentration of chlorophyll A in the lake is more predictable. If we use random forest, the R square is about 0.7, but it's still it's something that we want to see because the lake is usually used for water supplies and entertainment spaces. So it's good to have a higher predictive accuracy. However, it's still very hard to predict the concentration of chlorophyll A in the stream. If we are using random forest, the R square is about 0.36. The major reason is that the string is always moving, so it's very hard to predict. But that is something that we should put more efforts on, because the water quality of the inland water highly relied on the water quality of the stream that entering them. Lastly, even though we can have a relatively higher accuracy by using both remote sensing data and some institute measurements, but we want to include the institute measurement in the future, because our goal is rapid and large scale harmful algal bloom monitoring. So we want to get rid of some water sampling. And so in the future, we will integrate a multi-modality sensing across scales, develop a model to estimate the harmful algal bloom given concurrent observation, and develop predictive model based on neural networks and lab environmental inputs. And thank you so much for having us today. We will be happy to return next year with more results. Here is the reference.
Thank you so much, Schwab. I'm going to offer a few questions from the question and answer panel. So this one arrived very shortly after you began speaking, and it kind of bridges the last two presentations. So I'm not entirely certain who it's addressed to, but I'll ask you, and if you have a good answer, feel free to give it.
Okay.
Stephen Gastar asks, what would it take to model the relationship between algal cover and drainage, agricultural drainage in particular? What about by houses on septic systems?
Oh, please wait a minute. Let me see the questions. Oh, can you say it again, because I kind of lost.
Again, it's Stephen Gastar, if that helps you find it.
Stephen. Oh, this one. To tell you tomorrow should be. Oh, for this part in our research, will be contained in our promising model, because we believe that as I mentioned before, the agricultural land may have some nitrogen leaching, and it may bring this nutrient to the stream flows. So it may the nutrient will be contained in the stream. And our missing model is probably to modeling how long the plumes will stay on the water, and the plume, there is some nutrient contained in the plumes. This is the part that I can think about that our project will be related to the agriculture drainage.
Great, thank you. Ed Weinberg asks Schwa, for the nutrient inputs to the model, do you differentiate between total nutrients and soluble reactive nutrients? For example, total phosphorus versus soluble reactive phosphorus.
In our proposal, we will like it, in our proposal we only write out total phosphorus, but for the SRP maybe we can consider it in the future.
Great. And then one last is not so much a question as a suggestion. Dr. Thomas Mace suggests you include temperature as a variable in your model.
Yes, thank you so much for this suggestion. We are also calculating the temperature data, but we don't have time to put it in this slide yet. Yeah, we are working on it. Thank you.
Great. Well, that's all the questions that we have in the question and answer panel for your presentation. Thank you so much for your time today, Schwa.
Yeah, thank you so much.
And now we have arrived at break time. So if you can stop sharing, Schwa.
Okay, let me see, how can I stop sharing?
There should be a red button in your Zoom toolbar.
Oh, yeah, thank you.
Thank you so much. So we're at break time. We will break for 20 minutes. We will resume promptly at 11:40 Central. During the break time, Zoom will remain open if you want to continue staying active in the chat. But I've just checked and there's no line at the bathroom. So feel free to spend 20 minutes indulging whatever indulgences you feel like indulging. We will resume promptly once again at 11:40 Central. Thank you for your time today.
Quick note, it's 10:40 Central, 11:40.
Oh, sorry, 11:40 Eastern, 10:40 Central. Thank you for correcting my time zone disability.
Not a problem, happens to the best of us.
One more thanks go out to our sponsors who you can see on your screen. And without further ado, let's get back into the presentations. So if you're here, Mira, I'm about to introduce you. So I'd like to introduce Mira Gopinath Sujatha. Mira is currently doing her PhD in computer science and engineering at the University of North Dakota under the guidance of Dr. Ranganathan. She completed her bachelor and master degrees in computer science and engineering in the year 2008 and 2016, respectively. She's had experience working with Accenture Services, Ltd, and iNet Labs in India. Her current research is focused on accurately predicting the formation of harmful algal blooms via understanding various influential factors. Her area of research also includes data science, Internet of Things, and wireless sensor networks. And I'm sorry for any pronunciation faux pas, but welcome, Mira. I'm eager to hear your presentation.
Thank you so much for the introduction. So, yeah. You can see my screen.
Looks good, thank you.
So, hello all. Thank you for the introduction. And this presentation is about multivariate factors assessment of harmful algal blooms in various lakes. This is my preliminary year of my research. And I would like to thank my advisor, Dr. Prakash Ranganathan and Dr. Scott Coram at the University of North Dakota for helping me, giving me advices in this work, as well as I would like to thank my teammate Devashi Patel for helping in this, who helped me in this work. So multivariate analysis is a statistical procedure for data analysis involving multiple variables, and there are several methods involved for performing multivariate analysis. So this can be categorized as dependence methods as well as interdependence method. So dependence method involve various understanding the cause and effect of various variable over other variable, like multiple linear regression and MANOVA, whereas interdependent methods is not understanding the cause and effect, but it used to understand the correlation exists in the data structure, data set. So understanding the various factors affecting the formation of blooms, there are several factors which can be categorized as weather factors, water quality factors, and which is again affected by nutrient runoff from like total nitrogen, total phosphorus, and so on. So, so there are several literature already available for which studied the various factors influencing the formation of the bloom using several techniques like principal component analysis, canonical correspondence analysis, and so on. So in this study, we used the data set from 20 reservoirs from the states of Ohio, Kentucky, and Indiana. And so each reservoir has having the data about cyanobacteria concentration and other environmental parameters from the year 1987 to 2018. So in order to understand the relevance of the factors which are more contributing towards the cyanobacteria, so we tried to form a region-wise analysis. Like, for example, some of the lakes we categorized in the north region, and some on the east region, some under south, and some lakes under west. So we are categorizing the data set into 4 separate region data. So the parameters used for our study include cyanobacteria, chlorophyll, seshi, total phosphorus, total nitrogen, alkalinity, and precipitation, surface temperature, and dissolved oxygen. So we followed a 3-stage approach. So, so the data preprocessing, here we apply data preprocessing step to categorize the data into north, east, south, and west. Because the initial data we got was actually each lake we had only a single data point for each year. So in order to perform a better prediction using multivariate time series, we have categorized the data set into multiple regions. So then we perform a feature selection method to extract the relevant factors, the first 4 relevant factors in each region data using principal component analysis and Pearson correlation. And then finally, we performed which factors we perform dependence method of multivariate analysis like vector autoregressive and Aremax model to predict the cyanobacteria concentration in each region. So the second step, feature selection, was performed using principal component analysis, which is a unsupervised feature reduction method. So it generates a new set of variable called as principal components. It will describe the variance of data with minimum error. So we applied principal component analysis and extracted the most highly correlated variables with cyanobacteria, as well as we applied Pearson correlation analysis and compared the results. So this was performed in R, and these are the steps we applied for extracting the principal component and understanding the correlation. The final output was a loadings plot for each region, and this loadings plot is for the south region data, and we applied this correlation for the entire 32-year data, as well as last 10-year data and last 5-year data. And we found that the correlation from the loadings plot, the positively correlated variable with the count is extracted. Similarly, we extracted the Pearson correlation. We analyzed the Pearson correlation matrix, and positively correlated variable from Pearson correlation is also extracted. And we did for the last 5-year data, last 10-year data, and last 32-year data. So, and as we explored each region data, we see that the cyanobacteria concentration, which is greater than 100,000 cells per milliliter, in most of the lakes, starting from the year 2013 to 2018, was very high in east, west, as well as north and south. So because of that, we took the factors which are more highly correlated with for the last 5-year data. So we took the factors such as chlorophyll, surface temperature, total nitrogen, and dissolved oxygen in North region data. And in East region, we took total phosphorus, surface temperature, alkalinity, and chlorophyll. So South region, chlorophyll A, like total phosphorus, surface temperature, and alkalinity. And in West region, we took chlorophyll, alkalinity, total phosphorus, and dissolved oxygen. So based on these factors in each region data, we then performed the multivariate time series method using vector autoregressive model as well as Aramax model. So according to vector autoregressive model, it will predict a variable's value based on its past value observation as well as by analyzing the past observation of its exogenous variable. So initially, we study the stationarity of the variables in each region. And as we already mentioned, so the number of lakes in each region was 5. So there are 5 data points in each year. And in the north, there were 3, and west 4, and south 8. And we found that surface temperature in east, chlorophyll, total nitrogen, surface temperature in north, total phosphorus in west, and alkalinity and surface temperature in south were non-stationary, and we converted these non-stationary variables into stationary by means of differencing. So the next step was next step was to find the optimal lag using statistical selection criteria like AKK information criteria. So we found that in the east, north, west, the lags was 1, and south it is 8. And it is very important for the WAR model to find the optimal lags. And then we perform a co-integration test to understand whether to build a WAR model or VECM model based on the data set. So we found that East region dataset and West region dataset, we applied WAR model by analyzing the trace and max eigenstatistics value. So if this test value is greater than 5% critical value and the rank is full, we applied WAR model. Otherwise, we applied VECM model. And in the East region with lag 1, we applied WAR model with lag 1, and we found that alkalinity is significant in the long run, and total phosphorus is more significant in the short run. So in the West region data, we found that total phosphorus and alkalinity are significant. So in the North region data, we applied VECM model with lag 1, and we see that chlorophyll and dissolved oxygen are significant. So diagnosing the WAR model using multiple tests is also performed. So we conducted root tests to test the stability of the model. Serial test is to understand the serial, whether there exists any serial correlation in the model. So, and ARC test is to test the volatility, and normality test to test the residuals, to check whether residuals are normally distributed. So East region, all the regions data set passed the root test. So East region passed the serial test, arc test, and root test. North and West region data set passed root test and arc test. So whether South region data set passed only root test. So the next important diagnosis which we applied was Granger causality and instantaneous causality test for the 32-year data. So Granger causality will check whether the past value of the independent variable affect the future value of the predictor variable, whereas instantaneous causality will check whether the future value of the exogenous variable will affect the future value of the predictor variable. So we found that in the East region data, alkalinity, the past value of alkalinity can help to predict the future value of cyanobacteria concentration, whereas the future values of chlorophyll, total phosphorus, and alkalinity can predict the future values of cyanobacteria concentration. In the North region data, the past value of chlorophyll, surface temperature, and dissolved oxygen can predict the future value of cyanobacteria concentration. Whereas in West region, the past value of total phosphorus can predict the future value of cyanobacteria concentration. So, and the future value of chlorophyll and alkalinity can predict the future value of count. And the future value of chlorophyll and surface temperature can predict the future value of count in the South region data. So impulse response function is another diagnostic tool which is used in VAR model. So it shows the prediction. So if a small change or shock from the variables can impact other variables. So for example, a small change in chlorophyll can have a huge impact in count. So this has been done in the East region. For example, in the East region, this is for one period. So one period is having in the East region, we are having 5 points. So, so we see an increase in concentration of count from the increase in chlorophyll. So as a total phosphorus, an increase in total phosphorus initially have a small decrease and then it is showing a gradual increase. Similarly, with the other region data set, we also see there is the graph has been shown. So we performed a Aremax model, which is a dynamic regression model with the same 4 region data. So Aremax is auto or regressive integrated moving average for this exogenous variable. So we also tested the stationarity of the variable using the same approach. And then we applied Aremax regression equation on all the region data set. And we find that there was an increase in concentration of cyanobacteria with increase in chlorophyll and total phosphorus in the East region. Then in the West region also, there we see an increase in concentration of concentration of count with an increase in chlorophyll, total phosphorus, and alkalinity and dissolved oxygen. In South region, we see an increase in chlorophyll concentration of count with increase in chlorophyll, total phosphorus, surface temperature, and alkalinity. Both models were evaluated using rolling forecast method. And we tested we checked the MAE value, MAPE value, and MASE values. And we found that North region data set is having lowest MAPE, MASE, and other error values. And moreover, VAR model showed better performance compared to Aremax model. So studying the influential factors using multivariate analysis will help to understand the cause of one variable over other variable. So the VAR VCM model provide a range of tools such as Granger causality test to help to understand which variable is contributing to other variables. So that's it. Thank you.
Thank you so much, Mira. So at present, we only have one question. Ed Weinberg asks, what do you think makes the West unique in regards to total phosphorus causality?
So total phosphorus, so West unique, so maybe the number of data points or the number of lakes I took for each region data that has contributed to so I checked with the 32-year data. So when we check for the if the data set varies, the results may vary. The results can vary.
Thank you. More questions are rolling in. So Heather Merritt asks, well, first she thanks you for your presentation. I agree, thank you. Do you have any plans to assess the presence of microplastics, possibly of varying sizes, as a model parameter?
Yes, yeah, I can consider that also in future. Thank you for that.
So Ronan Selby-Curran asks, will reducing chlorophyll levels in harmful algal blooms help to reduce blooms in various water bodies, or be an effective way to do so? That is, potentially limiting sunlight by shading a water body, etc.
Sorry, I didn't get, what was the question?
I'm not sure that your model quite gets at this, but feel free to give some random thoughts if you'd like. Will reducing chlorophyll levels in harmful algal blooms help to reduce blooms in various water bodies? Be an effective way to reduce blooms in various water bodies? Potentially, for example, potentially limiting sunlight by shading a water body.
I'm
not quite.
I'm not.
Go ahead. Go ahead.
At this situation, I am not sure because so this work is basically based on available sample data collected. So this was just an experiment analysis which I have done using the data which I got. So more work we need to be done in future with more data sets. Yeah.
Janice Cook asks, how much does alkalinity vary in the north, west, and south? Why is it only a key? Does it only seem to be a key variable in the east?
So alkalinity in east, that's what. So we see that as a 4 separate data sets. So so we applied the WAR model on 4 separate regions. So alkalinity might in the east. That's what does we see that east region data. So the north region data is having the lowest error rate. So mostly the north region data shows better results compared to east region. So
great. Arthur Isaac asks, any explanation for the difference in causality predictors across the regions? For example, why is phosphorus important in west, alkalinity in the east, etc.? Why different causes in different places?
Yes, that I need to explore more on that.
Thanks. Irma Leapart asks, is the Great Lakes region considered east or north? Can you explain more about alkalinity to specify, is it basic or acidity that is impactful?
So the regions, so these are some of the reservoirs which I considered. So in the east, we have these lakes from near to Ohio. So
I don't see in your range map, I don't see that Great
there are
Lakes
no,
sites were
yeah,
any sites directly on the Great Lakes.
yes.
Can you address the issue about alkalinity? So is it base or acid status that is impactful?
So this, I can't answer right. I'm sorry, I need to check.
That's quite all right.
Yeah.
John Gatto asks, isn't there a possibility of confusion between cause and effect when using chlorophyll as one of the variables?
So chlorophyll, why I chosen this chlorophyll, these factors are because by understanding the correlation study based on my second step, like I conducted a correlation study. So on each region, I found the relevant factors. So chlorophyll was one of them. So so in the east region, I found that chlorophyll was there in all the region data sets. So that's why. Yeah.
Great, thank you.
Yeah.
Ryan Sparks asks, are there any predictions for the Midwest region?
No. Only I took the regions from Ohio, Kentucky, and Indiana. The data was from 20 reservoirs from this part.
Thank you. Stuart Dyer asks, Mira, how could you apply the model in real time when the prediction is based on a priori knowledge of future water quality parameters?
So I remember this VAR model can predict can perform this Granger and instantaneous causality. So instantaneous causality says that the future values of alkalinity, for example, if we know the future value of alkalinity, that can predict the future value of. So we need to have some historical data prior, and we need to perform an analysis before on that lake, and then we can apply that instantaneous causality.
Thank you. Ray asks, how do you differentiate stationary from non-stationary variables?
So stationary, by means of this, I performed the test by means of augmented Dickey-Fuller test and KPSS test and box lunch test, and checked the results which produced true. Augmented Dickey-Fuller test produced true. Then it is stationary. Otherwise, it is non-stationary.
Thank you. Ed Weinberg says, I believe there are approximately 15,000 lakes in the United States impacted by harmful algal blooms. How many total number of lakes make up your data sets?
So this is only 20 reservoirs. So the 20 reservoirs from the from these states. So the data set I took from the already existing data in the study.
And that said, can you envision a practical way to incorporate data from more lakes and perhaps broaden the coverage of this model across a much larger region, across wider parameters?
Yes, yes. My future work is exploring multiple lakes using using satellite images as well as other data. I would like to explore more on that. Yeah.
Stephen Tuarto asks, cyanobacteria themselves contain chlorophyll. And so even in the absence of true algae, chlorophyll will always correlate positively. And so can something like chlorophyll actually show causality?
Yeah, yes, that's true. Chlorophyll should have the causality, but yeah, I don't know why it's not showing. Like in the north region is north. Yeah, instantaneous causality is for chlorophyll is there for east, west, and south, but it's not showing the causality from the Granger causality. Yeah.
I see. And I might have missed it, but did you also consider phycocyanin, which is a pigment that's only made by cyanobacteria?
Yeah. So the existing data set doesn't have information related to phycocyanin. Only cyanobacteria concentration was there, but in future I will consider that also.
Great. And our last question, Ray asks, as these are reservoir levels managed? If so, how might this be considered? So, for example, I know a lot of Ohio's reservoirs, including some that you sampled, are flood control reservoirs with extremely variable water levels.
Yes. So these are the reservoirs, yeah, from this reference I took, like these are managed by, I guess, like US Army Corps of Engineers. Yeah.
Great. Okay. Thank you, Mira.
Thank you.
And if you can stop sharing, please. Thank you again, Mira, for your time.
Thank you. Thank you.
So I'm going to introduce the morning's last speaker. Ibrahim Basari studied water resource management and agrometeorology at the Federal University of Agriculture, Abeokuta, Nigeria, for undergraduate studies. Basari is also a graduate of the Erasmus Mundus Joint Master's Degree Program funded by the European Union Commission at the University of Bologna in Italy, University of Cadiz, Spain, and University of Algarve, Portugal. Graduated with a double master's degree in water and coastal management and environmental management and assessment. Ibrahim is currently a PhD student at Clemson University in the US and focused on monitoring harmful algae blooms in freshwater bodies using machine learning algorithms. So Ibrahim, I'm especially excited to hear your presentation. Please have at it. While you're bringing up your presentation, a quick reminder, if you have any questions for Ibrahim, please type them into the Q&A function of Zoom. Go right ahead.
Thank you. Can you see my screen?
I can, although I'm seeing presenters mode, so I'm not seeing the presentation as I should.
Okay. How about now?
Now I'm seeing the presentation as though you're working on it, not the presentation itself. So I'm seeing the entire slide deck.
Sure. How about
There
now?
you go. That looks like I expect. So thank you.
All right, thank you. Thank you, everyone, and thanks for having me. My name is Ibrahim Busari. My presentation today is on chlorophyll A prediction as index of herbs using a deep learning model. Most previous panelists have talked about machine learning in one way or the other. So I'll just be going deeper into the machine learning aspect, which is the deep learning model. My PhD advisor is also among the audience, that's Dr. Deb Sahu. I'm currently a PhD student in the Department of Agricultural Sciences. So as a form of background, we all know herbs as a very global problem for freshwater and also in marine environment. So here in South Carolina, there has always been different issues from the Department of Health and Environmental Control, talking about the issues of herbs and how it can be managed and prevented. Or also, just as a form of introduction, we have different monitoring approaches, the in situ and the lab approaches, where you have to go to the samples, to take some samples, to analyze some samples and try to make inference based on the analysis. We also use the process-based models where we use different physical numerical models to predict and understand some of the concepts and the relationships between different water quality parameters and how it affects harmful algae blooms. Then we also have the remote sensing approaches. Thanks to previous presenters have talked about using different satellite images just to observe and monitor various herbs related instances, just to be able to monitor and predict and also try to just have an idea of what's going on with the parameters. Then we have these data-driven models, especially these days, thanks to the sensor availability. Now we have different sensors that measures high frequency data. We also have sources with this computing technology. We have different data-driven models now. So just for my research, I'm trying to use a form of data-driven models called the deep learning models. So deep learning models is just a part of the machine learning models where you give a bunch of data to the model, it learns patterns in the model, and use the patterns learned to now predict or monitor herbs in the future. I'm just using this model now to just answer two questions. That is, what is the effect of data frequency? That is, how does observing my data probably daily or monthly or weekly, how does it affect chlorophyll A predictions? And also, does it really matter if we try to monitor our herbs just using daytime observation, or if we use the full data observations, that is all the data available for the day, or better still, we just use our nighttime observations. So specifically, I'm trying to see what is the effect of daytime observations on chlorophyll A predictions. My chlorophyll A predictions is used as the index of herbs here because of the importance of chlorophyll A in for algae, because chlorophyll A is an important characteristic of most algae. So the study area I'm working on is actually in a lake here in South Carolina. It's called the Boyd Mill Pond, as about a water volume of 3,108 acre feet. So the idea of just like a form of historical, like a background on this study area is that there is a particular lake which is somewhat downstream. So the idea of having this, of studying my Boyd Mill Pond, which is this, is actually to be able to monitor whatever is happening upstream. So if you monitor whatever is happening upstream, you're able to prevent or manage whatever is going downstream. So that's just like a brief idea of why we're actually monitoring this. So on this particular water body, on this lake, we have this profiler that is installed on it. It has a YSI multi-parameter sensors that takes readings almost every 15 minutes. So the sensor has been installed since 2014, and it takes readings every 15 minutes. It takes different water quality parameters. You have the pH, the conductivity, the temperature, the specific conductivity, the dissolved oxygen, about 7 to 8 different parameters, and also takes the chlorophyll A concentrations too. So for the first objective, which is trying to see how the daytime observation, how the data, the sampling period, how it actually affects my predictions. The first is to acquire the data and also pre-processing it. Like I said, we have about 15 minutes, every 15 minutes, we have the sensors that collect this data. So we have about 150,000 observations for each parameter, which is immense for my data-driven models. So I use this to collect the data, then you pre-process it. Of course, there are some missing values that you have to deal with. You have to clean the data appropriately. For the pH, for the temperature, for the chlorophyll A sensors, you have to clean the data and all. Also, in addition to the chlorophyll A that was obtained from the sensor, we also received some graph samples of chlorophyll A from the laboratory. But then the issue is now that we will have about 150,000 observations from the sensor. We only have about just 120 graph samples because we can't really match the proficiency of using sensors to, sensor has to take the data every 15 minutes, but for the lab, probably it's just weekly or monthly. So there was a limitation there because we want to use the actual chlorophyll A concentration as our index, as against using the chlorophyll A that was obtained from the sensor. So we had to use some Bayesian regression, which is just trying to see, okay, what if we use a particular linear regression, which is the Bayesian method, to actually see if we can obtain a continuous chlorophyll A, such that we'll have 150,000 observations as we have as 150,000 observations for the other parameters. So we have to use a Bayesian linear regression for that. Then we resample the data into daytime, nighttime, and also the full data set. The full data set meaning that we use every data. Then the daytime means we use data from 6 a.m. to 6 in the evening. And the rest of the data is classified as the nighttime data set. So the idea of this is just to see when exactly is chlorophyll A concentration is expected to actually be higher, or when exactly do we expect the algae productivity to be high, which is during the daytime, which is, we assume we'll just to take it as 6, also checking different literatures. We use from 6 a.m. to 6 in the noon for the daytime. And for the nighttime, we use from say 7 p.m. overnight to the next day as our nighttime observations. Then we move to the LSTM model. Like I said, the deep learning model that we used was called the LSTM model. It's called the long short time memory model. It's a form of machine learning model, only that the significance of this kind of model is just that it uses previously observed information. It considers previously observed information before predicting what is going on next. That is, it goes back few time steps, depending on the settings, to just see which of the probably previous day or previous week data do I need to forget before I run my model. That's just the idea of the LSTM model. So later I would go further to explain actually how it works. Then after building the LSTM model, we try to see if we can quantify some of the uncertainty. So like I said, this is just trying to show what the LSTM model looks like. So it has different, not to bore us with this whole mathematical formula. So the whole LSTM, what it does is that it has 3 different gates, like an imaginary gate, let me put it that way, called the input gate, the output gate, and the forget gate. So the input gate determines which of the new data that is coming today do I really need to predict my model. Then the forget gate determines which of the previous observations that I have in me should I just forget, should I ignore while predicting my model. And the output gate now determines which of the data that I have now do I want to use as the output of the model. So to put it into perspective, it's just like saying, let's assume I tell the data to use 7 days predictions, and I put a new data today, the model would go back 7 days time and determines which of these 7 days data do I need to predict my model now. Then after doing that, it now determines, now that I have my results, which of these current results do I need to output for as the prediction of my model. So for this model, we use the pH, the conductivity, the soft oxygen, the temperature, and some other about 7 to 8 physicochemical parameters was used as our input. And the output was the chlorophyll A concentration that I talked about. Now the derived chlorophyll A concentration that we obtained from the the Bayesian model. So now, the first result is about the Bayesian model. Just like this is a Bayesian linear regression, which is a bit different from the frequentist linear regression, that is the normal linear regression that we know. In that, what we're trying to do is try to see from our 120 lab obtained chlorophyll value, can we try to repopulate it into about 150,000 observations that all our other water quality parameters has. So what we did was that we found the corresponding day. For example, let's assume that for the first lab obtained values of chlorophyll that we have was today, we obtained the chlorophyll, the corresponding sensor value for chlorophyll A for that day. So we're able to find the same 120 observations for sensor, 120 observations for the lab value too. Then we try to use a Bayesian regression to just repopulate. So we obtain a model using the Bayesian regression. So after obtaining the model, we now use that to now convert the whole other sensor chlorophyll that we have. So that way, using the Bayesian model, we're able to have different parameters instead of the normal regression model that just give us, that will give one particular value for slope, one particular value for the intercepts, and one particular value for the error. We have different particular value. It gives us like different parameter value. So the trace plot is also showing us the vector of the samples that was produced using the Markov chain Monte Carlo sampling for each parameter. And also the kernel this distribution is showing us the marginal distribution of each parameter that was chosen. That is, instead of having one parameter for the slope, we have different parameters. In essence, we're able to have about 150,000 chlorophyll A concentrations that would be used as the output of our model. So that way we have 150,000 observations for all our inputs, and we have 150,000 observations for all our outputs. So this is like the first result. That is, we try to use the full data set. That is all the data set that we have. We use it to predict our model. And what we did was that we divided in the model such that 70% was used to train the model and 30% was used to test or validate the model. But this was done in such a way that we consider the temporal dynamics of the data set. That is, we didn't randomly train our model. We told the model that use 2014 to 2017 to train the data set such that the time sequence is actually considered. Then use 2019 to 2021 to validate the model. That way, we're able to keep the time dependence of our data. That is, instead of just randomly selecting 70% of the model to train the model and just randomly select 30, we didn't do that because we're trying to see if we can forecast in the future and such that we actually keep the time sequence of our data. So just like this is just trying to show the result of the testing data set. But we realized that although the trend of the chlorophyll A concentration was actually covered, especially when we see the upper boundary, because it's our model, because we use a form of LSTM model that enables us to actually have the lower and upper boundary of predicted chlorophyll A. We're able to see if the observed chlorophyll A is actually captured between this particular band. So we realized that at some point, although the trend was actually captured, we're able to see some spike that was not covered. Like here, we can see I spike here that was not covered by our model. So this can be related to probably measurement uncertainty, because normally data-driven models you might actually cover the model uncertainty, because when we are trying to train the model, when we're trying to find the optimum parameters, we're trying to find the best model parameters so as to reduce the uncertainty of the model as much as possible. But then little is being done about the measurement uncertainty, because anything could have been wrong with how the data was actually acquired in the first place. So this could be related to probably why this was actually done. Also, it can realize that it's just a spike, which could actually be like an error from the measurement or whatever. It could have been wrong when the data is actually being collected. So we actually related that to the reason why we have some spikes that was not covered, even although we check the moving averages, we realize that the upper boundary actually captures all the peaks very well. In the same vein, this is also showing our daily observations too. It shows the pattern, it shows the same pattern with the daytime and also the nighttime, also showing some spikes as we said in the first one, which was not covered. But then we could actually, just like you say, we related this to the measurement of certain, and also due to the fact that sometimes when we train the model, for example, like you said, we consider the timeness of the time sequence of our data. So the problem could be that the training data set could probably did not actually capture the real range, because we are only training it with a particular time period. Something could have happened in the next period that was not covered in the training data set. That is, the distribution of the data set could have changed, and it wasn't covered in our training data set. But then this could be improved probably due to if we have more information about the data set, or we have more data set, probably more features is being added to the model. For example, because for now we didn't include the nutrient data set or some, if you have the total nitrogen or the phosphorus, maybe this could have been improved, which is what some of these really focus on now, and also see if we can also deal with the measurement of that we talked about. So this is just now trying to show us the performance using the root mean square error. We use the root mean square error, we use the mean absolute error, and also the correlation coefficient. But this is just showing the root mean square error and trying to see how it actually changes. So this sampling period, that is, when we use the full data, when we use the daytime, and when we use the nighttime, we have different dropout. Your dropout rate of 0.1, 0.2, 0.5. Dropout rate is actually a feature in the LSTM model that is just used to minimize the overfitting of the model. Most from literature, 0.2 is actually most used as the dropout value. So we kind of focus on this 0.2, and we realize that our RMSE, the root mean square error, was actually lowest when we have full data, which is actually not so surprising, because when you have full data means you have enough data, which gives the model enough time to learn before predicting. But then you have to consider that this model, the more the data, the more the memory they consume, the more the computational time and all. So the second objective, what we are trying to say, if we resample the data into daily, that is, instead of using daily observations or hourly or 6 hourly or 12 hourly, does that actually affect our model predictions? That is, instead of me saying I want to be sampling every hour, I want to be sampling every day, I want to be sampling every 12 hours, is there really a significant effect on the prediction of my HABs using the chlorophyll A concentration as the index of HABs? So the difference between the first objective and the second objective is just how did we actually resample our data? For the first one, it was done for the full data set, the nights and the daytime. For this one, we resample the data into the daily, the 12 hourly and the 6 hourly observations. So that was the only difference between this model and the previous ones. So what we did was that we resampled the data so that we have the daily, the 12 hourly and the hourly data set. Then we now use this to fix our model. Still the same way we use the LSTM model, and we also use our pH, the conductivity, the specific conductivity, the soft oxygen, the oxidation reduction potential, the same thing as the previous objective. The only difference is just the resampling method. So we use this to now predict our chlorophyll A concentration, which is used as the index of our HABs here. So this is showing the result. It shows that we realize that when we are dealing with the daily, when we are dealing with with 30 days, we're trying to see what if I'm trying to predict my HABs 3 days in advance. Which of my data set, the sample data set, would actually be more effective? So we realize that if we are trying to predict 3 days in advance, using the daily data set is actually more effective. That was I'm only showing the daily data set because it was effective. We also use the 6 hourly, we use the 12 hourly, we use this the hourly data too. So this is just showing that the daily data set was actually more effective when we are predicting my chlorophyll A concentration 3 days in advance. But when you're not trying to predict 7 days in advance, we realize that when we are predicting 7 days in advance, we realize that the hourly data set was actually more effective. This is not really surprising because when you're actually predicting 7 days in advance, you need more data sets. That is, you need finer resolutions so that your model would actually learn effectively, which is why it's not so surprising. because we need more data if you really want to do 7 days in advance for your prediction. So trying to see now, this is just trying to show the same path error matrix using the RMSE. Like I said, we also use the mean absolute error and the correlation coefficient, and it also shows the same pattern. Like I said, for the 3 days prediction, we actually saw that the daily data set was actually the most effective. And when you are trying to predict 7 days in advance, the hourly presentation was actually better because it gives the data, it gives the model more time to actually learn before actually predicting for the future. So just trying to show the main findings, just that we realized that the full data set actually performed better when we used our conventional 0.2 dropout value. Also, we realized that the daily resolution data set was actually more effective when we are trying to predict in 3 days in advance. But then when you're trying to predict for like 7 days in advance, if final resolution is needed, which here the hourly resolution is actually providing it's actually more effective for that particular prediction. So it's just trying to show that ML, machine learning based models, can be used for predicting, and in the long run can be used for developing real-time decision support systems. So like I said earlier that we have problem with the measurement uncertainty now. Now we are trying to see if we can quantify the measurement uncertainty itself. That is, can we now kind of join both the measurement uncertainty and the model uncertainty just as to improve our model? Also, we're also seeing if we can just introduce the data assimilation concept so that we're able to learn the data very well, we're able to understand the data very well effectively. And finally, we also see if we can also use this same model after validation we can extend it to micro-cysteine and also some other cyanotoxins, which could be actually more detrimental to the health of the ecosystems and to the lives of the living organisms and all. So finally, because we obtained the data from the Greville County Department of Land Development and also the CCIT Clemson here, because we actually use the high processing computing from the institution to run our models and how. Thank you.
Thank you, Ibrahim. We have one question at the moment from Hyping Eye, who says, I think additional quantification index of the model performance by R2 is necessary. And how long is the time of predictions that you can make by the LSTM model?
So I didn't quite listen to the first part of the question, but for the second part of the question, I think it all depends on it's user dependent most times. So, and like I said, the more time you give it to predict, the more time it consumes to make the predictions. For example, if you're trying to say the model should do probably 90 times in advance, you know that it's going to take more time than just predicting 2 days in advance or 3 days in advance. So I don't think there's a particular limit. It all depends on the computing power of the system you're trying to use to predict.
Great, thank you. My own question is, we've seen this used in models a couple times today regarding chlorophyll. Of course, lots of organisms produce chlorophyll A, but only cyanobacteria produces phycocyanin. Do you think that collecting phycocyanin data, whatever is available, might change the utility of your model?
Yeah, of course. I think I quite agree with you. Including the phycocyanin could probably impact the model. I mean, it could go both ways. It could actually improve the model or actually not improve the model. It depends on the actual release, just like we said, most of these systems are very complex. So it depends on actually what exactly is going on. Probably there is also another, another confounding variables that is actually influencing the phycocyanin or the chlorophyll concentration in the system.
Great, thanks. And this question came as I was asking mine. It's tightly related to mine, so I'm not sure that you will have any more to offer on this, but I'll read it anyhow. Kale Clausen asks, how does your model distinguish between harmful and benign algal blooms when it uses chlorophyll A?
I don't think it's actually, I don't think the model would actually, because it's only predicting chlorophyll A. It doesn't know if it's harmful or not. But the idea is just that, so for example, here in South Carolina, the limit for chlorophyll concentration is if it is 40 microgram, if it's above 40 microgram per liter, that then something is wrong. So if we have, say for example, for a whole week, we have above 40 microgram per liter, the Department of Health and Environmental Control would probably say it is harmful. So because it's a limit for the state. So this could actually be state dependent, I would say, based on probably based on different regulations. But I think the model will actually say to actually identify if it is harmful or not based on chlorophyll concentration. I don't think the model is actually trained to do that.
Great. Thank you. We have no more open questions at this time. Thank you, Ibrahim, for an excellent presentation.
Much.
And I would like to thank all our speakers for their excellent presentations. You'll find their contact info on the screen right now, so feel free to drop them a line if any of your questions remain unanswered. We are to break for lunch. While we break for lunch, the Zoom webinar will be closed. We will reopen the webinar. We will reconvene for the afternoon session when we will discuss harmful algal bloom detection and treatment at 1 p.m. Central. That's 2 p.m. Eastern. Thank you so much for being here today. Thank you to our sponsors. It's been a great day so far. I'm looking forward to the afternoon.